So today we have two presenters, Snehal Patil and Raymond Cavalcante, who are both members of my lab. But this is actually a project that I am collaborating on with Aller Karnofsky. And so both of our labs have been working on this for quite some time. Some of you may be familiar with ConceptGen, which was based on genes. This is, I don't want to give any preconceived notions, but it's a somewhat similar tool, but with a lot of differences, and so you'll hear about that. And so Raymond will start off with kind of the motivation and what it's all about, and then Snehal will present some of the back end, how it actually works. So I'll give it over to Raymond.
Thank you. All right. Hello, everybody. Thanks for coming. So I'm going to be talking about ConceptMetab, which is a way to explore relationships between sets of compounds. So,
talk a little louder.
Oh, yeah, thanks. I tend to like mumble. So just to get it started, what is ConceptMetab? It is a tool that maps tens of thousands of compounds to biological concepts that are drawn from a number of databases, which many of you might be familiar with, such as KEGG Pathways, Enzymes, Gene Ontology, and medical subject headings. And I have a little asterisk next to the mesh terms because those are unique to ConceptMetab relative to other metabolite set tools. So in addition to mapping metabolites, we also map compounds that are shown in the literature to have biological activity. And so from your perspective, it's a web application that you can use to explore the relationships between the sets of compounds that we've annotated. So to get started, just what are metabolites? They're just small molecules that are in our bodies and in other organisms that are chemically transformed during metabolism, and they're necessary for us to exist, basically. And metabolism is just the set of all of the enzyme-catalyzed chemical reactions that sustain us and other organisms. So it's been said that metabolites serve as direct signatures of biochemical activity, and so in that sense, they're easier to correlate with phenotype. So they're kind of interesting in that sense. And I've just provided some examples. L-arginine is just an amino acid. ATP is kind of the energy currency of our body, and acyl coenzyme A is used in fatty acid biosynthesis very often. So what is metabolomics? It is kind of the characterization of all of the small molecule metabolites that are found in cells, tissues, biofluids, and organisms. And it investigates the products of gene, protein, and environmental interactions. And so it gives information that's complementary to genomics, proteomics, and transcriptomics. And so a lot of effort has been made into mapping these metabolites to biological functions. And so I've just pictured kind of the hairball of KEGG Pathways. And this is kind of one example of annotating metabolites and compounds to biological functions. So to give you a sense of what ConceptMetab is, I want to describe for you the mappings that we've done. So at top are kind of our concept types, I'll call them. And so we have medical subject heading terms, which come from the National Library of Medicine. It's a controlled vocabulary. We have KEGG Pathways, which many of you are familiar with. We have enzymes, and then we have GO terms from the Gene Ontology database. And so I'll kind of step through how we mapped each of these kind of higher level biological functions to eventually compounds. So to start, I'll tell you how we map mesh terms, which comprise kind of subsets, or it is comprised of subsets that are anatomical. You can have chemicals and drugs. You can have diseases, which is particularly interesting in a lot of cases. Organisms, phenomena and processes, which kind of overlaps with GO terms. You can have psychology and psychiatry, and then kind of technology, industry, and agriculture, which is kind of like environmental type stuff. So in order to map mesh terms to PubChem compounds, we go through a tool which was also developed here called metab2mesh. So metab2mesh was developed by Maureen and Alla, and Alex, and basically what it does is it associates PubChem compounds with mesh terms. So every PubMed, so it looks through the entire PubMed article database, and associated with each PubMed article is are mesh terms and substances. And those chemical substances can be linked to PubMed substances. And then essentially a Fisher's exact test is done, which associates whether a compound occurs in a paper along with a mesh term or not. And so we build our mesh term metabolite sets based on all of the compounds that are associated with a particular mesh term. And this is kind of novel, a novel annotation between compounds and mesh terms. So the next database that we use is KEGG Pathways, and so this is actually rather straightforward because you can just get KEGG Pathways as XML files, and then contained in the XML files are just all of the compounds that are in that KEGG pathway, so this is rather straightforward. The more difficult annotations to make are with enzymes and GO terms, and so what we end up doing is going through KEGG Pathways. And so I've kind of highlighted in this KEGG pathway of pyruvate metabolism kind of two compounds that are in circles, and then an enzyme which is actually linked to a gene in the rectangle. And so in the KEGG pathways, these two compounds are linked by a reaction which is catalyzed by this enzyme which has a gene associated with it, right? So from this, we can pull out that there is a gene associated with these two compounds. That's kind of the first stage. And then, in order to relate enzymes or GO terms with the genes that we've just related to the compounds, we use just an bioconductor package called org-hges.db. And then we get all of the mappings from GO terms or enzymes to genes, and then we go through all of the KEGG pathways to map all possible genes to all possible compounds that they are connected to through a reaction, and then from there we get all of those annotations. Okay, and so if you kind of step back and look at all the mappings that we've done, we kind of have literature-based mappings which comprise all of the mesh terms, and then we have pathway-based mappings that are really going through KEGG on the right-hand side. So we kind of have two types of annotations, and the intersection of them can be quite interesting. So after we do all of the mappings, we first require all the metabolite sets to have more than 4 metabolites in them and less than 4,000, so that we have A, enough statistical power, and B, enough biological specificity within our metabolite sets that we're comparing. And so this is just a table that gives you a sense of how many concepts we have annotated per concept type, right? And so you'll immediately notice that the mesh terms have a lot more concepts on average, and that they also tend to be much bigger. They have more compounds in them per concept than the GO terms or the KEGG or enzyme pathways do. Not only that, when we look at the mesh terms, we annotate order of magnitude greater more compounds to these biological functions than we do using GO or KEGG. So that's just something to keep in mind. And so in the end, we're going to be interested in figuring out the relationships between all of these metabolite sets that we've created. And so in order to do that, we're going to look at the overlap of compounds between all the metabolite sets. And so we were curious how promiscuous are compounds within the concept types, essentially, and within the concepts. So this is just all of our concept types that we have. And on the x-axis, we have the number of concepts that a compound belongs to on the log2 scale. And so what you kind of notice, if you look at the mesh terms, you kind of see that they are left skewed, which indicates that a lot of the mesh terms have compounds that are unique to only certain concepts, only unique to one concept. But they also tend to belong to more concepts as well. So you kind of have these mesh terms that have very unique compounds, but they also have very promiscuous compounds. Whereas if you look at the Go terms, they're kind of much more willing to share, on average, their compounds with each other. So this kind of plays into the association tests that we're going to do. So in order to, oh, right. So this is just looking at what are the concepts or the compounds that are most common to all of the concepts. And what you'll notice in this list is a lot of energy-related compounds like ATP, AMP, ADP. And that kind of makes sense because these are kind of necessary for all of the reactions to take place in all of these biological processes. So it makes sense that they occur in a lot of concepts. So if we're going to look at, if we're going to be comparing metabolite sets that are based on PubChem compounds to metabolite sets that are based on KEGG compounds, we have to have a relationship between the KEGG and the PubChem IDs. And so kind of the next thing that I'm going to note is we need a dictionary that kind of tells us that ATP in KEGG, which has this ID, is the same thing as ATP, which has a different name in PubChem, but has that PubChem ID. And so the idea is if, well, it's actually quite nice. KEGG has an API where you can just have all of the connections between KEGG IDs and PubChem substance IDs, which are slightly different than their compound IDs. And then in the Metab to Mesh database that was previously developed, we are able to map the substance IDs to the compound IDs. So basically, we have this link through KEGG and Metab to Mesh that lets us know which KEGG compounds are PubChem compounds and vice versa. So that way we can do association tests between all of our metabolite sets. And so we're just going to use a modified Fisher's exact test, which counts the appearance of the metabolites in two concepts, A and B. And so if you're going to do a Fisher's exact test, it's important to know what your background set is. And in this case, it's just the intersection of the background sets for the respective concept types that the compounds, or that the concepts are from. And so we just do this FDR test, or this Fisher's exact test. And after we do like 300 million of them, we compute the FDR because we need to account for multiple tests. So once we do those 300 million tests between all possible pairs of metabolite sets, it turns out we get like 30 million or so pairs that are FDR less than 0.05 associated with each other. And so this is just a heat map that indicates the level of association as a percentage of tests done between each pair of concept type. And so kind of what immediately jumps out at you is the metabolite sets that were based on KEGG compounds tend to be more highly associated with each other than those that are associated with the mesh terms. So they kind of like pull apart. You'll also notice that there tends to be a lot of enrichment along the diagonal relative to off the diagonal. But kind of the interesting biology is kind of between the concept types. So that's like a nice place to look and explore for interesting relationships. Right. And so with that, Snehal is going to talk about how we developed all the software. Oh, you don't need this.
I don't need this. Hello, everyone. Thank you, Raymond. I'll be talking about the software development part of the ConceptMetab. It's actually a tool which is deployed on the website. Oops. Okay, so these are the topics I'll be covering. Mainly, ConceptMetab is designed or developed using Grails, and it has a backend database MySQL. And we are using different charts or networks to show the data in the form of pie chart, networks, and heat map. And lastly will be the demo. So, Grails. We have used Grails to code main logic of ConceptMetab. And Grails is kind of a dynamic framework for web application. And it is an advanced version which is based on Java, Spring, and Hibernate. It is a MVC framework which encouraged the separation of concern. Now, what is MVC framework? It has like different compartments where you define the different definitions. Like model will have all the data object definitions. View will have how the user will see the pages, like JSP pages or HTML pages. And the controller will have the actual application logic of the project or a tool. That is kind of a link between the system and the user. So that is the MVC framework in Grails. Why we chose Grails for ConceptMetab is it has the scaffolding feature. What does the scaffolding does? It analyzes our model object and it creates the controller and views for itself just with one command. So generate all will create the controller and views for your domain object. So basic applications like create, read, update and delete, these kind of different pages, these will be created in just one command. So that is one good advantage. It allows you to do development very faster because you don't have to worry about the basic application. Another advantage of Grails is the plugin system. So it's like apps. You can import those apps into your tool and you can add the additional functionality into your tool. So for example, ConceptMetab use one of the plugin called export, which will allow you to download the data in different file formats like CSV, XML, or like even Excel format. So that plugin feature is also important for ConceptMetab. We have used MySQL as a backend database. Now the MySQL it doesn't have like a very commercial features like other commercial softwares or databases has. But most of the operations we are doing on the database is going to the database and just retrieving the data. Most of the processing is done using Rails. But for that purpose, MySQL was the faster one, and that served the purpose. Along with that, it's secure, inexpensive, and it manages memory very well. We can keep both the web app and the database on the same server, and that allows us to faster retrieving the record. This is the overview of our tables. Like concepts, there are like around 24,000 plus concepts which are distributed in 12 different concept types like mesh, KEGG, as Raymond mentioned. And there are like 89,000 compounds which have either PubChem ID, KEGG ID, or both, and remaining tables. So basically the workflow for the ConceptMetab system is when the user comes to the web browser, based on the query of the user, it will go to the ConceptMetab server, which is basically a Grails. And then it will go, based on the query, it will go to the database and it will fetch the records or data from the database. And it will send it to the server, and server will create the visualization pattern like a network. Now, if user has a specific requirements like the heat map, then in that case, ConceptMetab goes to the R server, creates the heat map, creates a matrix, and send it to the server, which will create the heat map for the user. So this is like a general web flow, how the tool works. Now, we are using different visualization pattern to create our, to display our data, like pie chart. So pie chart is basically a Google API, which is a simple JavaScript which you embed into your website, and you load your libraries and pass your data to that library. In our case, we are using JSON object to pass our data. And then you can customize the results using, like this is the snapshot of the customization parameters. You can hard code the parameters, or you can give, or you can change the dynamically parameters. Like for my case, concept types changes different according to the concept we have selected. So the color of the slice will change accordingly, and we can change that during runtime. So that is one good advantage of pie chart, that it doesn't have to be hard coded. We can change it during the runtime. So this is how it looks on the website. It basically shows the distribution of concept types of all the concepts which are highly enriched with the concept of interest. And then you can select the database from here and click on update chart, and it will update your pie chart during the runtime. Next is the networks. So we create basically two different types of networks in ConceptMetab. First is a star network, and other is a complete network. And these are created using Cytoscape Web. So this is modeled after the popular Cytoscape software, but it doesn't have the plugin architecture that Cytoscape software, which is a downloadable software, has. So it's just a reusable component which allows you to embed that code into your HTML document, and you can create the different networks. It is based on Flex and ActionScript, not on the GRE, which is a good advantage of Java Flex, because it doesn't depend on the Java versions or a security issue we are facing right now. So, and in case of ConceptMetab, node represents the concept, and their size and color are represented according to their number of compounds and the type of concept types. Similarly, for edges, they represent the significant enrichment, and their thickness depend upon the number of overlapping compounds. And we have used a force-directed layout to display the networks. So this is a simple network or a star network where the concept of interest is at the center, and all the other concepts which are enriched, they are around that main concept. And you can change the different parameters here, like p-value and odd ratio here, and you can change their effect, you can see their effect on the network. Similarly, for complete is a combination of star network plus all the enrichment relationship among the concepts. So yeah, okay. The next and the last visualization pattern is a heat map. So for heat map, we create that in R using the Heatmap2 function from the gplot library. So basically, we are creating a cluster, a matrix in Heatmap, saving it, sending it to Grails, and Grails creates the image based on the size of that matrix. We had some problems because of the size of the matrix, so we create two different views for the matrix. One is the table view, which is for less than 300 compound or concept size, and more than 300 will go to the image view, which is a static image created by Grails. The cluster is created by hierarchical clustering, and we use the Euclidean metrics with the average linkage criteria, in which rows are concept and columns are compounds. So this is the simple view. When the concept and compound both are less than 300, we use this table view where this image is created by R and this is created by Grails and the JavaScript. So we can scroll around this table and see the different values which are presented into this image. And another is the image view. This is a static image, which is a zoomable image, and you can zoom in and zoom out of this image and see the results. Let me show you the demo part of it. Why not? Okay, but it won't go there, right?
I think you need to mirror it now.
Okay, so this is the main web page of ConceptMetab, where it is deployed right now, conceptmetab.med.umesh.edu. There are two ways to explore ConceptMetab. First, you can explore by concept or compounds. I'll show you the compound. So if you have particular compound in mind, or you want to look for a particular compound, you can start typing in the name of the compound you want to look for, and it will show you all the possible names in the database. You can select any one of the compound, and it will show you the list of similar compounds. Okay, so this is the display page where this is the name of that compound, KEGG ID and PubChem ID. These are connected to the KEGG and PubChem website, and also the KEGG structure of that compound. Now, if the KEGG ID is null, then we go for a PubChem structure and we show the PubChem structure. This number of concept indicates to how many concept that compound belongs, and it also lists all the concept below.
Now, you changed the thing, and so it's now no longer broadcasting.
Oh,
You did that.
should I?
If you take it back, then you can see it.
Should I just extend it, or?
How does she do that?
Mirror it out on there, but then you'll have to like.
Okay.
Move that one.
Oh, let's see. But it doesn't go there.
Does it go on the other one?
Okay. Does that work? Okay. Okay. Now, if I have to search for a particular concept in this, I can just start typing in here like this, and it will show me the concept I want to look for. Now, I can go to concept page from here, or I can go to the main page and type in the name of the concept I want to look for. For example, like this one, I'll select the concept and then the name of the protein. Oops. Okay. So this page shows the information name, and in case of mesh, it shows the tree numbers and the concept type and the number of compounds that concept has. So if you click on this number of compounds, okay. So if you click on the number of compounds, it will list all the compounds that concept has along with their PubChem and KEGG IDs. And you can also download the results using this one of the, okay.
Snehal, in the order there, is that just a random order? Is the order based on the strength of the association, or?
For the compounds, it's just order, right? You can adjust the different parameters like p-values and odds ratio here, and that will change the pie chart which is displayed here. For example, this. So this will update the pie chart down below, and it will also update the list of the concept types which that concept belongs to, or concept of interest is enriched with. So you can select any one of the concept type or select all the concept type and go to the different views which are available here like star network, complete, or a heat map table. So this is the network which is created when you click on the star network, where the protein of interest, concept of interest is at the center. Something about the unfolded protein response. It is the well-known cellular response which occurs during the time of stress, and it has been identified that it is connected with the endoplasmic reticulum stress. And if you see that, you can easily identify that in here like this, and it is also connected with the cell death, which is this. So you can identify the relationship which are actually published or which are already linked or established with the help of star networks. Now what this star network has is this concept, or the node represents the concept. When you click on that, it will show you the information of that concept, like what is the name, what is the concept ID, and how many compounds it has. The size of this node changes according to the number of compounds it has. For example, like diet high fat, it has less compounds as compared to the cell death. That's why the size is smaller. Similarly, the edge thickness depends upon the number of overlapping compounds. So if you click on the one of the edge, looks like it's a bigger one. Okay, so 16. It will show you what compounds are making that intersection and what are their PubChem and KEGG IDs. You can zoom in and zoom out this and even the move network around. And this is using Cytoscape Web. You can also change the different parameters here and see the effect of that parameter on your network. From here, you can go to complete network to see the results, which will have the star network plus other enrichment associations. And this is similar to the star network. The nodes are represented by concepts and edges are overlap. And then, okay. Okay, it's
Snehal, can you explain the dotted lines?
Yeah. Yeah. This is the subset superset relationship between the concept. If one concept has a compound which are belongs to the other concept also, like that is a subset of that concept. So that is a subset superset relationship. The arrow indicates which one is the superset, and the other end is the subset. Okay. You can go to the table view from here, which will list all the concepts along with the link to see which are the overlapping compounds. For example, this one. You can see different compounds which are there, and you can also download it from here. You can go to the heat map to see the results. This is a general overview of how heat map will look. And if you want to see the detailed structure of the heat map, you can click on this one. But after that, there will be different views depending upon the concept into compound matrix. If it is a smaller matrix, it will go to the table view. And if it is a larger matrix, it will go to the image view. So this is the table view. And here is the table which you can scroll down and up and see the results.
Is there any way to export that?
For the table, we have the unclustered file, which is at the start of the page, but not this table. You can download the image, but not the table right now. Okay. So here is the link to download that file, which you can cluster it into R. Okay, let me show you the image view also for the, I'll need to add some concept for the larger matrix. For example, oops. I guess, yeah. You can update the chart, sorry, I forgot to mention that. And you can go to the heat map. The heat maps are a little slower because it involves going to the R, coming back from the R and creating it image. Okay, so this is a static image which is created using Grails, and you can zoom in this image, or like if you want to look for this part, you can zoom in this image and see which are the compounds and concepts are connected. So this is heat maps. Okay, now let me go back. Okay. Is it already there?
Close it and then like restart the presentation.
Okay, so this is the future development. We are trying to add the experimental data into the ConceptMetab website, and if you have any suggestions, this one doesn't work now. Okay, if you have any questions, let us know. Any questions? Any suggestions?
I know map was PubChem with KEGG. So for the directory mapping, is there any way to see the structure similarity? That is, they have exactly the same structure then different.
Yeah, we were thinking about do.
Like multiplied or
Yeah, that's a helpful thing to look at.
I
We
think
haven't.
there are software for chemical similarity.
Yeah, we were thinking about adding metabolite sets that were based on chemical similarity also. So yeah, or we could use it to map that. That's almost.
Questions?
Sure.
I have a general question. Was there any reason why you use like KEGG instead of PFAM, or like can I use the GO and the KEGG for basically in the beginning? Is there any reason why you chose that instead of PFAM? Like even for pathways instead of like KEGG pathways instead of like meta pathways or something. Like you just
Well, PFAM doesn't really have metabolites. They cluster proteins, right? So if you're thinking about Reactome maybe or other databases, well, KEGG is, the most commonly used database.
Yeah. I guess we didn't do a thorough comparison, but
Yeah.
I think KEGG annotates the most metabolites. We wanted to have much
in terms of metabolic clusters.
even as you saw with KEGG, it's only annotating a couple thousand compounds, whereas with mesh we got more than an order of magnitude more than that.
And I mean, I think some of the, Raymond, correct me if I'm wrong, but I think some of the tools that you've mentioned at the beginning that do this sort of thing looked at other databases like Reactome, for example. For sure, I think they're mostly redundant with KEGG. So it's a good suggestion, and we should probably look at it. But so KEGG seemed like a good place to start. I think.
Yeah, and we didn't want too many redundant pathways. But mesh is really the novel part of this, because the other related applications, well, nothing is really like this with exploration. But did you mention the MSEA and those? No. Okay. There are a few programs that will try to do metabolite enrichment with experimental data, but so few metabolites are annotated anywhere in them that a lot of times they don't give you any real results. So what we're trying to do with mesh is try to get a much larger percent of metabolites to be annotated somewhere.